we're fortunate to have John back for a second time. There's obviously, if you read some of the information, you know the subject matter, but it's really interesting to me that John has developed with his partners a method of reducing the amount of damage that the drugs that we normally use to try to attack cancer do while trying to attack the cancer cells. So from a very pedestrian perspective, it seems like a logical approach. So we're super happy to hear more about what John is doing and some updates from the last time that we spoke. So don't forget to put your questions in the Q&A or the chat box, and we'll get to as many as possible. And as you see today's presentation, if there's anybody that comes to mind in your community or your network that would benefit from seeing this, you can review it on the Family Office Insights YouTube channel, the recording, and also feel free to contact John directly. We'll make sure everybody has the opportunity to do so. So with that, welcome back, John. Thank you for doing this today. Super, super interesting stuff. And so take it away. Thank you.
Thank you very much, Arthur. Thanks for having me back. And thank all of you for your interest in attending, for being here, for being present, and being interested in the technology I'm going to tell you about. And my name is John Hall. I'm the Vice President of Research and Development at a small biotech company located in New York City. That's Molecular International Research Incorporated. I'm going to tell you today about an investment opportunity based on our US patent titled with this number here you see on the screen, and there are other worldwide pending applications. The subtitle I've given is Non-Toxic Anti-Cancer and Anti-Inflammatory Drug. The actual title of the patent is Selective Anti-Cancer Agent, Effective for Prevention and Treatment. And of course, I'm giving this for Family Office Insights, and thank you for having me back. Before I go right ahead, I want to make two quick points. The first is that just yesterday, I heard that in addition to the US patent, we now have the patent in the EU, the European Union has been allowed. This is a big plus for us. That's a large market and there's a lot of activity there. So not only does it increase our market, but it also increases opportunities for finding partners. There's a lot of development that goes on in Europe, and we've learned from our own experience that we need to have the US patent to proceed there. So with both EU and US, I think we're in good shape, and there's some more that are pending. And I think that's also good for potential investors. You can see that we're expanding the patent portfolio, and it's promising and indicates our success in the intellectual property arena. The other thing I want to mention, which is the take home of the whole talk, I think what we have here, and Arthur alluded to this, reducing toxicity is the key. But if we can develop this drug, we have a high impact change in the way cancer treatment is conducted. A non-toxic drug, and I'll be expanding on the potential of it, is a high impact development. And I know many investors in all fields, but I've seen, especially in cancer, there's a desire to see that their money's going into a project that, if successful, will have a big impact. And that's exactly what we offer. Well, let me begin with a quick summary of my talk, describing first what I'm going to tell you, and second, what we're asking for. So we have, as I just said, researched and patented an effective non-toxic anti-cancer drug. So the drug itself kills cancer cells, but it itself is not toxic to healthy tissues and organs in the body. The new drug works against most cancers, so it has broad spectrum, including the most difficult to treat, pancreatic and lung. And we focused on those two in particular. All our evidence, everything we know, predicts that the drug will be safe in the human body. So I'm going to show a little bit of information about that, but we have more. And for those who are interested, by all means, let's talk about that because it's not wishful thinking that we're not toxic, that our drug is not toxic. It's not a hope and a prayer. It's real. So the drug also functions as an anti-inflammatory, which is opening a second large market. I'm going to be talking a little bit about that. Taken together, the many advances of the drug make it an outstanding investment. So what we're asking for, with a goal of licensing or acquisition by the pharmaceutical or larger biotech industry, we have to bridge the gap from where we are now to human clinical trials. So that will cost us $3.5 million to fund this milestone. It's a transition to human clinical trials from patent to humans. And it's we're seeking an investigational new drug application that will be approved by the FDA. It is that IND, once it's approved, that enables clinical trials in humans, which are essential for FDA drug approval. So completion of the IND research also opens opportunities, more opportunities for partnering with the larger industries we're focused on. So let's go back to what problem were we trying to solve when we got into this? And that is, simply put, chemotherapy drugs are not selective for cancer. So cancer cells grow quickly, and chemotherapy kills fast-growing cells. Chemotherapy also kills normal cells that are fast-growing. Death of normal cells causes serious side effects. Patients suffer, and treatment must be stopped before therapy is complete. So this is an industry with this built-in flaw that's been there for decades, because the drugs, while they kill cancer cells, also do real harm in the body that accounts for the negative side effects, which can be so serious they force suspension of treatment and ultimately have an effect on contributing to the demise of the patient, quite independently of the cancer itself, but often in conjunction with the cancer. What is our solution? What did we do? Our newly patented drug, diethylflavopirin, selectively destroys cancer cells, including cancer stem cells, in a solid tumor or a site of metastasis without damaging healthy tissue. Diethylflavopirin can be used as cancer therapy for many different cancers. So it's a broad spectrum anti-cancer drug, but it's non-toxic in the animals we've studied it in. The diethylflavopirin can be administered orally and is well tolerated even at large systemic doses. It can also be used as an anti-inflammatory. I want to emphasize the oral administration, which is by far the best route of administration. It avoids all the complications and costs of intravenous and is much easier on the patient, particularly suitable for prevention of cancer, not just treatment, but it also works for treatment orally, but also for the anti-inflammatory activity that we have. So this actually answers a lot of questions. So let's take a look at the drug itself. And this slide is yes.
Could you jump 2 slides back?
Sure.
So I don't know how you attribute data to this, but just intuitively, we know that the lifestyles are impaired dramatically by chemotherapy, right?
Yes.
So one would assess from hearing you talk about this that I think I remember you saying that this can be administered in conjunction with chemotherapy and often is, if you anticipate it being, but that if we just looked at it from the perspective of how it could be less debilitating being in chemotherapy, is that realistic that the lifestyle would improve?
Absolutely, yes. And one of our arguments is to work together with the industry that is established for so long, not to simply come in and say, well, we can take over and solve everybody's problems. The chemo drugs are here, and most of the scientists and oncologists I talk to say they're here to stay for quite a while longer. Even though there are new forms of therapy like immunotherapy and targeted therapies, which show promise, serious promise in some cases, The old drugs are still with us. And so one of our goals to enhance our acceptance among oncologists and clinicians, and even in the pharmaceutical industry, is to enable, is to have shown that our drug works together with these traditional mainstream treatments, which are toxic. Ours is not. And I can tell you that we do see not just a big benefit in overcoming the chemo resistance. One of the problems with chemo is also the cells become resistant, the cancer cells become resistant, and they stop responding to the drug. And one of the things our drug clearly does in some cases is revert the cancer to chemosensitivity, meaning the chemo drugs work again. There is also in there the promise, the beautiful promise of a dose reduction effect. So that directly addresses your question, because if you combine a chemo drug with our drug, you need less of the chemo drug in order to reach an equivalent toxicity than you would if our drug was not there. So you can actually ease up on the toxic agent while you have ours there, which kills cancer cells itself, but also reverses the chemosensitivity and the resistance, chemo resistance, and can then lighten the side effects. If you have this dose reduction, the dose reduction refers to being able to reduce the dose of the chemo drug that's toxic.
Yeah, thanks.
And that has a real future if it's taken seriously.
Sounds promising. Thanks for sharing that.
Yes, thank you.
Continue. Thank you.
So let's look at the molecule. I entitled this Improving on a Natural Molecule. And the one that's synthetic and patented is on the right. It's called diethylflavopiririne. You will see that it is quite closely related to another molecule which occurs naturally called flavopiririne. They both have this tetracyclic, 4 cyclic structures together. The difference is this ethyl group, which is shown in red on the left. That group hangs off the lower side of this ring, and that's what we removed. We said, let's see if we simplify the naturally occurring molecule, which of course cannot be patented, cannot be used. But one advantage we had was knowing that flavopiririne, through numerous studies, does show anti-cancer activity. And we said, how about taking that step where we make an alteration, in this case, simplify, and come up with a molecule with similar structure, and we hoped similar activity, and see if anything else changes, just see what else we find. Well, what we found was that the diethyl, our synthetic molecule, when the ethyl group is removed, the anti-cancer activity is retained. To our surprise, we found that toxicity is reduced relative to flavopiririne. This is the naturally occurring compound that's actually found in the bark of a tree that grows in the Amazon rainforest. So that was the reduction of toxicity was a surprising result at the time. And we realized what a great result, because that's really what made our molecule open to patent allowance, because this really has a functionally distinct character from this naturally occurring molecule. It's less toxic. I should say that flavopiririne to start with is not very toxic. That's low toxicity to start with. So I think here we've taken a step towards what I refer to as vanishing toxicity. And in the animals, we've used it in by oral administration using large doses. The doses are well tolerated, and the tumors shrink, and the animals appear to be just fine. We see no detectable side effects. So this is a great development. It's an unexpected result that really helped us get the patent. And in a sense, I like to say we've improved on the natural molecule, which, believe me, doesn't always happen when chemists get out these molecules or make a plan to make changes or alterations in the structure. It's not uncommon to find there's greater toxicity in the molecule that's been changed. Here, it's quite the opposite. And another way to put that is that the tetracyclic part of this accounts for the anti-cancer effect. The ethyl group does not confer anti-cancer, but it does bring along a little extra toxicity that our molecule does not have.
Would it,
so just, yes.
Would it also be logical that this will be synthetic, and so you could reproduce it at a much more of it to be very you probably
Absolutely. You can
so
make as much as you want.
yeah, that makes sense to me. Yeah.
And that's really important because the world will demand a lot of it if it gets out there and gets used. So diethylflavopirin, the molecule I've been talking about, is referred to as NT1391. So 1391 refers, which is what I'm going to call it, refers to the sulfate salt of the compound, just one of the natural forms of a salt that can stabilize it. NT stands for non-toxic. We were so impressed by the lack of toxicity that we put that in the name of the compound. Okay, let's see. What then is the result of giving this compound to an animal, or ultimately it'll be a person who has cancer? What we found is that in cancer cells, treatment with NT1391 upregulates genes associated with apoptosis pathways. This leads to cancer cell death by suicide. And that's important to reiterate. Built into our cells are pathways that lead to a cell self-destructing. That's called apoptosis. And it's a wonderful avenue to go down with a cancer cell. The problem is doing it selectively, which is what we can do. I think many of the other drugs, of course, don't do that. They're killing off the healthier cells in some cases just because they divide rapidly. Our data in animals shows that 1391 induces apoptosis in pancreatic cancers and in colon cancers with no detected toxicity in animals. Recent research shows that flavopirin, some of it's done with purified flavopirin, there's some labs studying that, and diethylflavopirin, which is 1391, use multiple signaling pathways to trigger apoptosis in various cancers. So P53, JAK-STAT, AKT, P38, and NF-kappa B among them. That's important to say because it's a strong positive aspect of this type of cancer treatment, of our method of treatment. Once the drug is in a cancer cell, it looks like it's going to die by one of these pathways, whichever one the cell actually uses to reach apoptosis. The cancer cell is going to die. So that's a strong benefit of this approach, this multiple pathways towards cancer cell death by apoptosis. So what are the reasons to believe here? I just gave you one, which is multiple pathways to apoptosis. And I've also mentioned that it's not toxic. And I've come to refer to this as the gift that keeps on giving. Keep thinking of ways in which that's so helpful to remove the toxicity from an anti-cancer drug. And one example is that it can redefine the whole concept of anti-cancer treatment cycles. The idea of treatment cycles is built in when you're using a toxic drug. You're going to go for a certain period of time, which is based on experience with that drug, the condition of the patient, the tumors, the extent of the cancer and metastasis. And then the drug is withdrawn. It may be a few weeks, sometimes less, sometimes more, but there is a respite so that the patient can recover from the negative side effects, whatever they are. But this also gives the cancer a chance to rejuvenate. And that's exactly what tends to happen. And often by the time treatment is resumed, when the patient's up to handling it and can confront the side effects again, by that time, the cancer has started to regrow. The tumor comes back sometimes more aggressively. So with these toxic drugs, you're forced into this cycling, which actually has a negative side effect of its own, because it brings back, it enables the cancer to get the rest from treatment, and then the cancer can come back and sometimes more aggressively. I think that this is amazing. If you treat with our drug, you can keep treating. You can treat until you attain your desired goal of shrinking the tumor to whatever extent the oncologist decided is essential. And that defines a positive result in treatment. You don't have to stop because the body can't handle the side effects. We haven't found any yet. So that's another reason to believe it's really remarkable to think about out of the mindset of toxic chemo drugs. You have a drug that can still be effective in killing cancer cells, but it doesn't have this toxic side effects, and therefore can be used more extensively. And if the oncology chooses, you can bring in another drug to supplement. As I've said, we combine very well with other forms of treatment, so that can be done as designed and intended. It's not forced to be interrupted. The treatment regimen isn't screwed up because the drug is toxic. We can avoid that, which it's a big step. So it's broad spectrum. 1391 kills cancer cells of all types tested to date, and it works also against hard to treat tumors like pancreatic and lung. Well, I mentioned that we studied in vivo pancreatic and colon cancers. We also have done now studies in cells and drug combinations with lung cancer, so we can back up the claim that it's effective there. And this broad spectrum is telling us something, and I'll allude to that in a minute. It's highly effective in drug combinations, as I've said, and restores chemosensitivity of drug-resistant cancers. So the ability to work with other cancers, to collaborate, if you will, I think is a great strength. And reversing chemosensitivity, meaning reversing chemo resistance, let me get that right, is considered to be an essential feature of the next generation anti-cancer drugs. That's really important, and we've shown we can do that. This molecule also functions as an anti-inflammatory, and here our research has focused on the potential to treat cytokine storm, and there's other inflammations as well. But cytokine storm, as many will know, is actually it's the reason why people with severe COVID-19 infections actually succumb and die. That's because their own immune system has been overstimulated and is persistently stimulated. The regulation that reduces the stimulation and then enables the immune system to go quiet is not there any longer. So it just keeps pumping out white blood cells that go to the site of infection. And when there's enough of them for long enough, they start damaging the local tissue, say in the lungs, for example. So while they're programmed initially to go after the infected cells, the virally infected cells, if there are enough of them that accumulate in the tissue, they start going after their own body cells. And this is a real problem. And I think our agent can have a very beneficial effect here. Sepsis is another example. These systemic infections, which get out of control, and the immune system is just going crazy. I think we can reach out and then and get that under control. I want to also say here that this anti-inflammatory effect is a positive side effect. If you're treating someone with cancer, the anti-inflammatory effect is also present, and it's valuable because many of the cytokines in the cytokine storm actually induce cancer cell growth. They promote cancer's advance. So it's another way in which it serves as an anti-cancer, because those cytokines that would help the cancer grow can be attenuated with 1391. It targets cancer stem cells to prevent recurrence and metastasis. Cancer stem cells are just a small subpopulation of cells in a tumor that are responsible for chemo resistance and for persistence, no matter what the treatment, and then out the other end, they regrow and metastasize. So they're the problem when it comes to cancer recurrence. Cancer comes back because cancer stem cells are there. They survive the treatment, and then they start to regrow and spread. That's also an important feature to have in a next generation drug. Then I want to spend a couple of minutes on this breakthrough advantage. It's the selective mechanism of action. There's a number of reasons for this, and I'll get into it. What is the molecular foundation of selectivity? I've said that our agent, it kills cancer cells. It leaves the healthy cells alone. What we've learned from conversations with a couple of drug companies is they want to know exactly what the molecular target is. It's not enough to say, well, it's non-toxic. Well, they want to know what is it targeting specifically that it works that way to enable that lack of toxicity? So what do we know? The data show that there is a cancer cell specific target for 1391, shared by most, if not all cancers, a potentially universal anti-cancer target. This we see from the broad spectrum, and it's also clear it's built into the lack of toxicity because it's cancer cell specific. It's not in, that target apparently isn't in a healthy cell. And that's why 1391 just ignores the healthy cells. It doesn't target them. Anti-1391 can distinguish cancer DNA from healthy DNA because the DNA in cancer cells is damaged. So this is the fundamental basis of selectivity at the molecular level. It's selective binding to damaged cancer DNA that initiates apoptosis. I hope that makes sense to you. Please let me know if there are questions. I've made a...
John,
Yeah.
if you don't mind, just quickly sharing with everybody how this came about. Like it's an interesting story, right?
Oh, I think very.
Yeah.
Let me get to the next slide and
Okay,
help
sure.
answer, I think. Because it's a cartoon which hammers, if you will, the point that the DNA in cancer cells is damaged relative to the healthy DNA in a healthy cell. And here's just a symbol that signifies a healthy DNA structure. There are two strands, the DNA strands, the backbone, that are in real life. You can see in my decoration at the side of the page is a better picture of two intertwined spirals that form the duplex. It looks a little more three-dimensional. But here I'm just using this to make the fundamental point that healthy DNA, the structure is intact. And in our schema here, that's what an intact structure looks like. The DNA is hammered by the attack of free radicals. It's oxidative reactions. We've all heard of oxidative stress, but this is a lifelong process whereby our DNA is chemically damaged and, in fact, structurally damaged. You come out the other end and you have, instead of the neat duplex of a healthy cancer DNA, of a healthy DNA, you actually have this damaged DNA, which is characteristic of cancer cells. The nucleotides are modified. Some of the atoms in the ring structures in the DNA have been substituted or deleted. The bonds are stretched. Some bonds are sometimes broken. So I've heard people refer to the DNA in cancer cells, that it's structurally a mess. And this also includes the bending of the backbone. So it's chemical damage to the bases, the sugars in the DNA, but ultimately this leads to the backbone being bent. And this has a profound impact on the structure of the duplex. And that's what we're looking at as the cancer cell specific target. And we believe that this altered structure is what makes it up. It's not just that there's damage in the chemical sense, but it's that changes the conformation, the three-dimensional conformation of the structure. Say, for example, at a replication fork, when the DNA is replicating, is being synthesized. And I think that's where we can get this cancer cell specific target. And I think that's where our molecule has a binding site. It targets that. So we've all heard about oxidative stress. I've been studying it a lot as I've started to see that it's oxidative stress that is the foundation for carcinogenesis. And that stress and the consequent damage are what our agent uses as a target. But it's also something to be mindful of in terms of our health. The oxidative reactions happen because of free radicals. There's plenty of those coming from the environment. Our own metabolism generates these free radicals that are highly reactive, and that's helped them change. It helps these free radicals change the structures I've been talking about in the DNA. It's chemical damage. So I think I also should say that this DNA damage is a well-characterized feature or aspect of cancer DNA, and that's been known for some time, starting with Mirko Belchanski's work and then really solidified and advanced by Don Mallon's work. This, for example, I'll give you one more example. He analyzed the DNA from a population of prostate tissues. And he can distinguish the structure of a young man's healthy prostate DNA from an older man's. And indeed, the older man's DNA, even if he doesn't have cancer, actually more closely resembles the DNA damage seen. There's enough damage there, it starts to resemble a cancer DNA. But that condition of aging, it's just part of this process. It's part of being on Earth and living what we do. And I think maybe it's made worse by all of the environmental influences we confront. But as I said, it also comes from our own metabolism. So one idea here is how important antioxidants are. And Mallon's looked at that. But to finish the point, from young person DNA to old guy's DNA, the structures are different. And then if you look at cancer DNA, it is another classification. It's another profile. And that also changes yet again if it's metastatic. So the extent of DNA damage is actually synchronized with the condition or the health of the DNA itself. And although this has been well established, I think we are the first to treat this damage and the consequent structural changes as an anti-cancer drug target. And it's partly because it's been known for a while and no one's really said, let's focus on that. Maybe that's a way to go after a cancer cell. And this is what we got into. And I think the results ultimately will speak for themselves. I don't know, did that answer your question, Arthur?
No, but it was pretty good.
Well, let me try it.
So no, but the question was more about how you came about discovering this patented drug.
Oh, okay.
So, but this was really interesting because for someone like me that doesn't, traffic in this business day in, day out, what you just described as something that's, while recognize that it damages the DNA structure, that they're attacking it with chemotherapy in a different way. So it was really interesting to hear that. But what I was getting at is the story associated with how you guys all came about discovering this thing.
Okay. Let me finish on this line in one.
Yep, of course.
This, although widely established, it's known that DNA has this damage in cancer cells. It hasn't been appreciated. And there is a prejudice there. The entire biomedical research community has been focused on mutations, which is changes in the genetic code. And that would show up as changes in the color of the bars here. If those change, it's a mutation. And what you've done is change the actual code of life. If that change occurs in a gene that codes for a protein that influences cell division, cell progression, and then has a role in cancer cell division and cancer progression, then you have accomplished your goal of finding a cancer drug target. But the idea that it's the DNA itself at this basic, what I call foundational level, really hasn't been much out there. There's been a real favoritism for the information model, that we're changing the code and that cancer is caused by that and it needs to be treated as a problem of a defective genetic code. I think we're showing that there's this other angle here, which is very, very important to pursue. Let me go back up here to this slide, which shows these two structures. Well, we were very much into flavopiririne because, as I said, it shows anti-cancer effect, and it's found in the bark of a tree that grows in the Amazon. And my CEO's father, Mirko Beljanski, Sylvie's father is Mirko Beljanski. He had a long career at the Pasteur Institute, and he was the one who identified this as an anti-cancer compound. And we've done a lot of experience with those plant extracts from the Amazon and explored the anti-cancer effects and shown a lot of mechanism of action for them. But that was really first and foremost in our mind, this structure. But we knew very well that it couldn't be patented. So if we wanted to explore intellectual property here and support making a drug, we needed to make a change to that molecule, as I described. And then we came up with D-ethyl, just removing this. And lo and behold, it then lowered an already low level of toxicity. And that is the story.
Yeah, that's what I was getting at. It was the history with Sylvia's father. Yeah. Thanks for doing
Indeed.
that. Yeah.
That was critically important in getting us into the study of this molecule in the extract form and seeing what it does. And Beljanski did a lot of experiments showing the anti-cancer effects. And in purified form, flavopirin is now currently studied in a number of labs, which continue to show that it kills cancer cells by apoptosis, in fact, which is the mechanism we find in 1391. So the advantage of our molecule is the lesser toxicity, but also that it can be patented and pursued as a drug.
Yeah. So, John, I think things in my own way. So just with that caveat, one would, in looking at this, say, well, if this works in its natural state, why isn't it being used? And part of the argument is that it can't be commercialized because it's not patentable, right? Not to mention that it may not be able to be scaled because there's just not enough of the trees.
That's key point here. That's absolutely key. Because this is available as a plant extract, and it is sold as a dietary supplement, meaning the flavopirin, right? The ancestor of the naturally occurring non-patentable one is out there to a very limited extent. There's a question of how do you sustain that natural source of this molecule in extract form. There's only so many trees, and there's only so many bark, only so much bark on those trees.
Yeah.
Which fortunately do regenerate their bark. So if you harvest it, it comes back. So it's very desirable, but it's not going to answer the needs that we have for cancer treatments of pancreatic and lung, colon. Yeah. It's also the need of the community of patients that need help from drugs like this, which from D-ethyl flavo, because they can take it and it's not going to hurt them significantly, let alone kill them.
Right. Yeah, super interesting. Yeah, I think about it from the commercial perspective, right? So,
yeah. Right. Okay. Here's DNA damage. So this is just, I have a few slides here to go over. This refers to the market. Our treatment answers an unmet need. Killing cancer cells selectively spares the patient the notorious toxicity of anti-cancer drugs. The expansive demand for a broad spectrum, safe and effective drug is reflected in our patent valuation that determined the fair market value of anti-pancreatic, anti-lung, and inflammation applications to be $799 million. Other common cancers, like colorectal, can also be treated. And there's a longer list. As I said, it's broad spectrum. Inflammation, NT-1391 can normalize the dangerous levels of immune signals in cytokine storm and in sepsis that cause tissue damage, organ failure, and death. I touched on this earlier. Many forms of inflammation can also be treated. So we tested alcohol-induced pancreatitis, for example. I think it should be said, for licensing and acquisition, inflammation may be separated from the cancer applications. Let me just show you one summary from the patent valuation, which was conducted by IP Metrics. Fernando Torres, the chief economist there, was extremely involved, and he got interested in this and really grew to appreciate the science involved and was able to come up with his valuation with that mentality. But at the same time, including at my insistence, we wanted just absolutely non-aggressive. We wanted assumptions that were conservative. And that shows up in the estimated market penetration. 14 to 16% for all thrmost applications, which is modest, but realistic. Lung cancer is worth the most because it has the most number of people with it. Pancreatic cancer, somewhat less prevalence. Doesn't have quite the value. Inflammation, somewhat less for what we calculated. I always feel that 799 million sounds like a lot, because it is a lot. But from what I've been saying, you will see that you'll appreciate that you can add a few more cancers. And what we had to do is focus on what really we think will work best for people and work in the market, be desirable in the market. So let's just see. Yeah, okay. Competitive position. Molecular International Research is the first company to research and patent a drug for cancer that is non-toxic, so our status is unique. This drug will initiate a revolution in cancer therapy, leading to drastically improved therapeutic outcomes for cancer patients and strong commitment from doctors who will prescribe a safe and effective drug. And it also functions as a non-toxic anti-inflammatory. And then here I come back to what I said at the very beginning, the first slide. The patent is allowed in the US, and we just received notice that the patent is now allowed in the EU. It's also pending in Canada, China, and Japan. I want to have a slide here that just acknowledges the risks of investment. And the fact is that cancer drug development has a low success rate of about 10% that actually reach market successfully. So, failures occur at human clinical stage, trial stage most often, up to 50% because the agent ends up lacking the efficacy. It's not active enough against the cancer. It doesn't shrink the tumor. It doesn't work. But about a third because of unmanageable toxicity. We don't have that problem. And then 10 to 15% because of poor pharmacokinetics. And we don't have that problem either. We've done pharmacokinetics. It's a preliminary thing. It's going to be redone in the IND research. But we're already well down from this, this categorization of failures, which I think we can improve, and we're going to stand up and we're going to look good in this against the risks.
Does this also explain why the big pharma, let me just call it that, would not be interested in throwing money at this or partnering until you got beyond into the human trials, simply because that part is so unpredictable in efficacy terms?
Yes, I think you have 50% because of lack of efficacy. And we see efficacy, there's no doubt about it, in animals, but you do have to, and you can extend that. It's predictive. So it should work in humans just as well. But you got to put it in humans and see what actually happens. You can't get around that step, and we don't want to, of course. And I think this came up the last time we spoke. If this doesn't work out, it's not because there's some known bugaboo or some problem. Basically, if this doesn't work out as a drug, it's for a reason which is just dropped out of the sky. We don't have an angle on why it would not work. And it's true that some anti-cancers that work in mice don't work as effectively in humans, but we did some controls for that. So we think we're in good shape.
Is there a realistic concern that it threatens the chemotherapy business?
We're arguing for inclusion in the chemotherapy business. But of course, it can be seen as a threat. It's not toxic. Think of the reaction to patients. And think of what these oncologists go through. The clinicians have to give these drugs to people, and they know there's a risk. They're doing the best they can, because there aren't non-toxic drugs available. So I think it is a threat to the status quo, and I've already described how we've tried to handle that by showing how well we work with the other drugs. I think that's the way to get acceptance. And once it gets into the people, they can decide how frequently to use it, and we'll see what happens. So just a quick highlight of the process of drug development. I want to focus in on what we're trying to do, what it's going to cost, and how we're going to spend the money. Typically, there's research done, hopefully innovative, that can lead to an invention. This is, in our case, a novel anti-cancer, anti-inflammatory drug candidate. Then there's a patent which details of the invention are described, then the claims covering composition and uses. It's a good time to mention perhaps that our patent covers, there are composition claims in it, and its methods of treating cancer. It is not a new chemical entity. We did not invent the compound 1391, so we couldn't patent it. And I think that's not much of a problem, but it's worth noting. The patent valuation is the start of a marketing phase, which we're in. I'm doing it right now. But the next step before you get to clinical trials, and then at the bottom, FDA approval for a new drug, is IND application to the FDA. IND stands for the Investigational New Drug, which you have to get that, which gives the FDA the toxicology studies, which I'm about to just explain briefly, and then the other data, pharmacokinetics. They need to decide if the drug is safe enough to put into humans. Initially in a phase 1, where the goal is to check safety, and then in more advanced stages as it passes each test. But this must be gone through. There's no avoiding it. And we have to get to the clinical trials, and then ultimately FDA approval of a new drug, which involves drug sales and establishing markets. So next step is the IND. As I said, the cost is $3.5 million. Approval of the FDA by the FDA for an IND requires safety to be determined in toxicity studies in larger mammals, including tests for impact on cardiopulmonary function. We've designed the metabolic and toxicology studies of the Charles River Labs. It's a world-renowned laboratory, well known for these kinds of studies. In discussions, the chief scientific advisor at Charles River said, if you're seeing efficacy for pancreatic cancer by oral administration, you're really onto something. And that's what we saw in our animal studies. The IND application, we have an expert we worked with before, Dr. F.A. Hoffman. She used to work at the FDA and has extensive experience with successful IND application. So this is a very detailed file that has got to be compiled, describing the manufacture, all the experimental work, the quality, the purity of the drug as synthesized, and how it's used. And all of that goes into a file that's then sent to the IND, which also then has to include the toxicology studies I mentioned. Okay, here's our collaborators. I mentioned IPmetrix and Fernando Torres. Compound synthesis is APAC Pharma. Worked with Dean Y for a number of years on a number of compounds. Reliable, excellent purity, a great place to have as a partner for chemical synthesis. The toxicology for IND, as I said, is Charles River Labs. Sam and Kyle are there. They've been extremely helpful putting this thing together. And then, as I mentioned, F.A. Hoffman of Heterogeneity LLC can do the application for the IND along with the FDA filing. The costs are set out here. This was increased somewhat in the last few months. I was checking them out to see if prices have gone up, and as we know, all prices have gone up for everything. And so there's a bit of an increase here. Compound synthesis is expensive, but we've done it so that if we make enough, we should have a lot. We should have enough for doing the toxicology, and may well have some left over. And if we do, that can be used in the first clinical trial because we're getting it made of a clinical quality. The toxicology is a big ticket. That's Charles River. There's some research folded in here, which is important. The mechanism of action I just alluded to is something that the drug companies want to see more data on, and we need to generate it and show it to them. And anti-inflammation applications, it's going to be possible to make some determinations about what other inflammatory conditions are really a good application for our 1391 drug. And then the filing, I also just mentioned that's listed, and some additional expenses. Please, anyone who's interested, you can go over these in more detail. So critical vindication of our safety profile in toxicology studies comes with the IND. Then we have mixed in metabolics. That's how is the drug metabolized in the body? What happens to it? It goes in orally. We've got to follow what's its fate in in the digestive system and then in the bloodstream when it gets there. Analysis of drug target interactions and optimization of drug delivery. The drug target is close to that, the mechanism of action, and whether we're really truly targeting the chemically, structurally damaged DNA characteristic of cancer cells. So when you get FDA drug approval, clinical trials, we have in mind 3 tracks. First, pancreatic cancer by fast track development, meaning there's no viable treatments for that. And so it's possible to get the FDA to help you accelerate the process to see if a new agent has the ability to help with that difficult cancer. Lung cancer, it may also be possible to do fast track, but we're not sure, but we're going to try. And then the inflammation application, cytokine storm. There's clinical trials that have been designed to study that. So here we have an FDA approval for a new drug. As I said before, it's not a new chemical entity. So finally, FDA approval will yield an anti-cancer drug with a new level of safety. Drug action doesn't depend on rapid cell division or specific mutations in a patient's cancer cells or on redirecting a patient's immune system. The drug will generate a new platform for drug combinations that will improve many forms of treatment. And in closing, I want to say a prominent pioneer of a new immunotherapy has explained how badly he needs a non-toxic drug that could shrink his patient's tumors by apoptosis so that his immunotherapy could become a more effective treatment. He might as well have been describing our drug, and many oncologists could say the same for their goals with their cancer patients. So I'm suggesting that our drug is something that won't just work with chemo, but with the newer forms of immunotherapy, for example, can be valuable with these other modes of treatment. And I think it represents what the cancer treatment industry really needs at this time. So of course, there's an urgency to move forward. Please let me show you our team. There's Sylvia Beljanski, our CEO. There's me there. She has an interesting background. Born in New York when her father was here on a research project at New York University Medical Center. She grew up in and was educated in Paris, studied history, and then went to the Pantheon to get a law degree and was admitted to the bar. I joined Molecular in 2004 to develop new intellectual property. Together, Sylvia and I have 3 successful patent applications, one for prevention and treatment of prostate cancer, a second for therapeutic treatments based on small RNA fragments, and the new patent, selective anti-cancer agent effective for prevention and treatment, is the subject of this presentation. It was a small company, it's privately held, there have been no outside investors to date, and we now seek investment for the IND phase of drug development. There's her background. I just gave a little synopsis there. I was born and raised in Princeton, did graduate work at Princeton University, and then finished up at New York University Medical Center. I was a postdoc at Rockefeller in New York, and then on the faculty. My work focused on microtubule organizing centers and microtubule organelle assembly. I served as a president and CEO of Motility Research Corporation, a biotech startup, and I'm currently vice president of Molecular International Research. Gary Tanagawa is the IP attorney who helped put our patent together. He and I work very closely, but he has a very good science background. He has a PhD and was a real help in putting this together, as you can imagine. But then he switched to add the law component at Georgetown University Law Center and works now patenting novel biotherapeutics for multinational, but also smaller life science companies. Joshua Goldberg is the newest member of the team. He's with Nav Goldberg and Meyer since 1997. He has a chemical engineering background from Washington University in St. Louis, and then went to George Washington University Law School. His practice is focused on both the acquisition of worldwide patent portfolios, particularly in the pharmaceutical and biotechnology industries, as well as assisting with his clients with licensing and commercialization of such products. So Josh is on board to help us do a deal, negotiate, and get something that works for the investors as well as for us. Now, I have some data to show, but I think we may not have time. Arthur, please let me know.
Yeah, so we've run out of time, but a couple of quick things. You've noted that haven't taken any outside investors into Molecular. how much money have you put into this so far?
It's over $1 million.
Yeah,
It's cost over now, probably well over since we last checked,
yeah.
adding everything up.
Right.
So that's where we are.
And this detailed discussion could probably wait, but if somebody just wrote a $3.5 million check, what's that buy? Does it buy into molecular, or does it buy into just this product?
I think right now this product is the value of molecular.
Fair enough.
Yeah, so I think it's going to be buying into molecular as things are usually done, but it's going to be based on this product specifically.
And is it the investment based on the estimated value of 799 million or some other negotiated amount?
Well, we have to let that happen in the negotiations.
Fair enough. Yeah.
I don't mean to put you off.
No,
but,
no, this
that's
is
a patent valuation, and there's
yeah,
lots of room for discussion
yeah,
in terms of what the valuation would be for an investment at this stage.
yeah, and that's what folks want to hear is, you know, where you stand on that, right? So, not the number, it's just that it's open for discussion. That's all. Yeah,
Absolutely open for discussion.
yeah, great. So yeah, we're totally run out of time, but this has been a great update, and I super appreciate you doing it. We'll make sure we get everybody in touch and socialize this with the community as we discussed earlier, and then we'll follow up with you. But that's been super helpful, John. Thank you.
Well, thank you so much for having me, and thank
Yep.
everyone for attending.
Yep.
Happy to talk to any of you.
Yep. Thank you, John. And as I always say, thank you for sharing with us the only thing you can't make more of, and that's your time. Till next time. Thanks, John. Really appreciate it.
Thank you.
Take care.
Take care.